Hello, let's continue with our discussion. And in previous lecture, we just talked about that there is a portal vein and there is a hepatic vein. They are going to the liver, and while blood is passing through the liver, that is blood and its content are exposed to the hepatocyte, and after that blood drains into hepatic veins and eventually into inferior vena cava and systemic circulation, right? So we can say this is portal circulation, this is systemic circulation, in between we have put liver. Now I put here one hepatocyte, and that hepatocyte I have drawn in a mega form here, in a big form here. So let's see here on the hepatocyte how our hepatocyte works on the glucose, right? First of all, let's suppose on this hepatocyte here is circulatory system and it is having nutrients. As I said, it is bringing nutrients, including it's bringing glucose, right? And it is also bringing insulin, C-peptide, incretins and other substances. Now, there are two things. First of all, I will talk about insulin, how insulin works. How insulin works. As you know that insulin is a peptide hormone. Because it is a peptide hormone, so it is long or big molecule. And of course, amino acids, many of them are charged. So it is big molecule and somewhat charged molecule. So insulin molecule cannot enter into membranes. Because insulin molecule cannot cross the membranes of the cell, the first thing we need to learn is that insulin receptors are present on the membrane, not within the cell. Why? Because insulin cannot enter into cell by dissolving into plasma membranes. Now let me draw the insulin receptor first of all and how the insulin system works, right? Insulin receptor basically consists of two alpha units, and attached with them there are these two beta units. Now alpha units and beta units, all of them are held together by disulfide bond. Let me make it here the big one. Here is the alpha unit and this is by disulfide bonds, this is held with the beta unit. Another alpha unit held with beta unit, and they are also attached with each other by disulfide bond. Now, this part of insulin molecule is extracellular and this part is passing through the cell membrane. This part is passing through the cell membrane. This part which is passing through the cell membrane, this is beta part. This part is also called transmembrane component of insulin receptor. So we can say insulin receptor has extracellular component, it has transmembrane component, and transmembrane component continues with the intracellular component. Now what really happens, that let's suppose insulin comes and sit over here, right, and it interact with the alpha units. When insulin interact with the alpha units are modified in such a way that they lead to modification in beta units. Again, when insulin bind with the alpha units undergo a transformational change which is leading to the further transformational change in beta units. In the beta units, there are special type of enzymatic activity. Yes, these stars are basically they are the integral part of the beta unit, but this star is basically having an enzymatic activity. What is this enzymatic activity? That they can attack tyrosine molecule, they can attack the tyrosine amino acids and put a phosphate on them. So these are basically tyrosine kinases. because these tyrosine kinase are integral component of the beta unit, because these tyrosine kinases are integral component of the beta unit, we say that beta unit have intrinsic tyrosine kinase activity. Beta unit has intrinsic tyrosine kinase activity. And now this tyrosine kinase activity, when insulin comes, alpha units are activated, beta units are activated, eventually intrinsic tyrosine kinases are activated. And what they are doing? Let's suppose here the tyrosine kinase, here is tyrosine, here is tyrosine, here is tyrosine. Now tyrosine is just an amino acid part of the beta unit. This, yes, this is very important to understand, that tyrosine kinase of one beta unit will attack the other one, right? And the second one, tyrosine kinase, will attack the other, each other. So what happens? Just imagine this is beta unit, this is beta unit, right? These are the two beta unit. So my thumbs are the tyrosine kinase, intrinsic tyrosine kinase activity domain. So this my thumb of right hand will attack the other hand, and here it will find the tyrosine amino acids and phosphorylate them. And thumb of this, the enzyme of this beta unit, will attack here and do the tyrosine kinase activity. So what really happens? In medical terms, we say that when insulin molecule is actually basically tetramer with two alpha units, two beta units held together by disulfide bond. When insulin binds with the alpha units, they lead to the changes in the beta unit, and each beta unit intrinsic tyrosine kinase is activated, and both beta unit attack each other and lead to the phosphorylation of tyrosine residue of each other. And eventually what we say that this is phosphorylation of beta units. So beta units intracellular components are themselves phosphorylated, and because they are cross phosphorylating, this is cross phosphorylation, so we call it heterophosphorylation. Once this unit is phosphorylated, then towards this phosphorylated, another phosphorylated beta units, another protein will come and bind here, and this protein is called insulin, yes, receptor substrate. What is this protein called? Insulin receptor substrate, right? So once the beta unit are phosphorylated, they attract the and hug the insulin receptor substrate, right? Insulin receptor substrate. And after that, yes, after that, these beta unit lead to attack this insulin receptors substrate and lead to phosphorylation of, yes, lead to phosphorylation of IRS. Once this insulin receptor substrate phosphorylated, who phosphorylated here? Again same beta units. First they phosphorylated each other, then both of them phosphorylated the incoming or upcoming IRS. Once it is phosphorylated, from here the pathway takes two different sides. One side is, okay, I will come over here now. Now you can tell me, what is this? Insulin. And what is happening here? Autophosphorylation of beta units. This is autophosphorylation of beta unit, but auto cross phosphorylation. Please don't use it hetero because it was a mistake. It is autophosphorylation of beta units. What was this? IR, insulin related receptor substrate, and eventually the tyrosine amino acids present over here, right, they are also phosphorylated, and in this way insulin receptor substrate activated after phosphorylation. Now it leads to many signaling pathways, but I will very briefly discuss two signaling pathways. One pathway is that it will bind with a special enzyme, okay, on this side. One enzyme binds over here, another protein binds over here. These are two pathways which are activated, but these two proteins are different. These two proteins are different. This protein is, yes, phosphatidyl inositol 3 kinase. What we call it? Phosphatidyl inositol 3 kinase. What this will be doing? It will look for a phosphatidyl inositol unit and lead to the kinase activity. What this enzyme is going to do? This is going to look for phosphatidyl inositol and it will add their third phosphate. Now, actually normally phosphatidyl inositol diphosphate are already present in the membrane. This will, it's a long tongue, this will act over this. Phosphatidyl inositol diphosphate and convert into phosphatidyl inositol triphosphate. This was the reaction. This will give intracellular signals. With multiple signals, eventually it will activate protein kinase B. This is also called AKT, protein kinase B or AKT molecule. Now, this AKT, that is why this pathway is called PI3K AKT pathway, right? Now, what this is doing? What this is doing? Actually, it leads to phosphorylation of many proteins, many different proteins, and what will be the result? Now listen very, very carefully. What will be the result of this thing? The result will be that it will, suppose multiple pathway, eventually it activate a phosphatase. Phosphatase. This is a phosphatase which is, suppose, phosphorylated by protein kinase B. This is phosphatase enzyme. It is quite possible this phosphatase is activated and it removes phosphate from different, let's suppose this is one enzyme and let's suppose this is another enzyme. Okay. This enzyme was having phosphate here and this was its functional domain of enzyme A, right? Now, when this phosphatase will attack here and remove the phosphate, the mouth of the enzyme will open and this enzyme will become active. So, it means that protein kinase B can activate many phosphatases which lead to removal of the phosphate and dephosphorylation of certain enzymes and protein, and when they are dephosphorylated, this unit will be exposed and become active. Is that right? Opposite to that, this become active. Let's suppose to this enzyme previously phosphate was sitting on the top right? Phosphates were sitting on the top and keeping its mouth closed. Now this enzyme, when it was phosphorylated, it was inactive. This phosphatase, if it removes these phosphates, this will open its mouth and this enzyme will start working. Mouth mean the active site of the enzyme. Active site of the enzyme. Now listen carefully. First we will make a very basic concept. I think we need to discuss basic concept first. Let's suppose these are two enzymes, right? Now, if you add phosphorylation at this point, right, do you think it can work? No. But if you if you dephosphorylate, it will start working. You phosphorylate, it will not work. So some enzymes, when they are phosphorylated, they don't work. This was the enzyme, when it was phosphorylated, it did not work. But when it was dephosphorylated, when its phosphate was removed, it started working. So we can say some enzyme in phosphorylated states are not working, and in dephosphorylated state they are working. But there are other enzymes. When they are phosphorylated, they are not working. But if you remove the phosphate, they start working. So we cannot say that enzyme is phosphorylated or dephosphorylated. It is sure to going to make it active or inactive. It depends, it varies from enzyme to enzymes. There are some enzymes, they work in phosphorylated form. There are other enzymes which work in dephosphorylated form. It actually depends on that how the phosphorylate acts and alters the functional domain of the enzyme. Is that right? Now come back to the insulin. So PI3K ACT pathway, right? This pathway basically gives signals, very complex signals of phosphorylations and dephosphorylation of different proteins. And those different proteins, if they are enzymes, then those enzymes undergo altered function. And when they undergo altered function, metabolic pathway will alter, right? So how insulin is able to alter so many different metabolic pathways? It changes the carbohydrate metabolism, it changes the fat metabolism, it even has effect on the protein metabolism. The answer is that this part of the signal transduction pathway, insulin, alpha units, beta unit, autophosphorylation, then phosphorylation of insulin receptor substrate leading to activation of PI3K, phosphatidyl inositol 3 kinase, which actually lead to convert phosphatidyl inositol diphosphate into phosphatidyl inositol triphosphate, which activate a special pathway which is called PKB ACT pathway, that is protein kinase B. This lead to multiple phosphorylations, and phosphorylations lead to even activation of phosphatases, and those phosphatases will dephosphorylate other enzyme. So it means what it is doing, insulin signaling pathway, that it is doing multiple actions on multiple enzymes almost simultaneously, right? That some enzymes are phosphorylated, others are dephosphorylated, some are activated and others are inactivated, right? This is clear. Now when enzyme activity can be altered by 3 ways. Number 1 is, let's suppose this is the enzyme, right? If I do here like this, or I put alteration here like this, so this is not phosphate, it is something else. Let us suppose it is substrate, right? If there is a substrate, come and activate it, then we call it allosteric regulation. What is this? Allosteric regulation. If substrate or some other substances which is not phosphate bind with an enzyme and influence its function, we say there is allosteric regulation. Is that right? For example, there are some enzyme, their function is altered by ATP, or altered by AMP, or altered by presence of citrate, or altered by presence of many different substances. So if an enzyme is altered enzyme function is altered by a substrate or by a product or some other, but it is not phosphate, right? Then it is called allosteric regulation of enzyme. But if phosphorylation occurs to the enzyme, if phosphorylation of the enzyme changes the enzyme function, we say covalent regulation. What is this? Covalent regulation. And third way to enzymatic activity alteration is increase or decrease the concentration of enzyme. So let me write here that enzyme functions can be altered by three ways. Number one is by allosteric regulation. Allosteric regulation, right, where a substance binds with an enzyme, right, but it does not bind covalently, and this substance binds with the enzyme and changes its function. This is allosteric regulation. But if there is phosphorylation or dephosphorylation, an enzyme function is altered, we call it covalent regulation, because phosphorylation and dephosphorylation makes a covalent bond with the enzyme. And third way to alter the enzyme function is, third way to alter the enzyme function is that either increase enzyme catabolism or increase enzyme synthesis. In this way, you actually alter the amount of enzyme. For example, if substance goes to a gene and increase the synthesis of enzyme, then that is called induction of enzyme. Induction of enzyme. Or if a substance acts on nucleus in such a way an enzyme producing gene is inhibited, we say suppression of enzyme. What we call it? Suppression of enzyme. It means enzymatic activity can be allosterically regulated, enzyme activity can be covalently regulated, enzyme activity can be regulated by alteration in genetic, you can say transcription and translation at the rate of synthesis of enzyme and catabolism of enzyme by induction or suppression of the genes. Is that clear? We come back to insulin and correlate it with insulin function. Here we have already learned that phosphorylations and dephosphorylations can affect the functions of enzymes. Now we come to this pathway. This protein with a long nose, okay? This nosy protein, right? We call it sons of 7, SOS. This interact with a protein which is called RAS protein. Sons of 7 act with RAS protein, and RAS protein act with special type of enzymes, and those enzymes are called, MAP kinases. MAP kinases. And MAP kinases give signal to the nucleus, and to the genetic system. So it means that once the IRS is phosphorylated, it can lead to two pathways. Number one, PI3K ACT pathway, which will bring rapid changes in the membranes, I will talk later, or rapid changes in enzyme function, phosphorylations or dephosphorylation. Other pathway is RAS-MAP kinase pathway. What we call it? RAS-MAP kinase pathway. This pathway actually affects the genetic material. So here it can alter the, it can alter the synthesis of enzyme, either increase the synthesis of enzyme or decrease the synthesis of an enzyme. Even it can alter the growth of the cell, it can alter the differentiation of the cell, right? Now, what we can say that we have learned that insulin, listen now carefully, insulin mainly act on the target enzyme by covalent regulation, by phosphorylations, dephosphorylation system, and by induction suppression system. They are controlling the enzyme activity by at the level of production of enzyme, right? Any question up to this? There's no question. Very good. Now, another thing which I want to say, this was about the insulin receptor, that now you know insulin receptor can give mainly two types of pathways. One pathway is going to work, this pathway work on the membrane and work on the cytosol, and this pathway mainly work on RAS-MAP kinase, mainly work on the nucleus, right? Alter the genetic expression of the cell. Now, let's remove it. Now we come to this discussion this was our just remnants of the insulin receptor transduction system left over there. Now we come to this cell, this is hepatocyte. As I told you that when from glucose is coming from the portal circulation, about 40% to 50% glucose can be taken up by the hepatocyte. But what kind of uptake occurs? Actually, hepatocytes also have the transporters, glucose transporters, and these glucose transporters are also GLUT2. These are GLUT2. GLUT2. As I told you previously, that GLUT2 were present on beta cells of pancreas, GLUT2 are present on hepatocytes, and GLUT2 are present on basolateral membrane of gastrointestinal system, and GLUT2 is also present on the basolateral membranes of nephrons, right? Now, hepatocytes membrane are having which kind of glucose transporters? GLUT2. Now, GLUT2 are not insulin dependent. Some glucose transporters are insulin dependent, right? They are insulin sensitive. Now, which glucose transporters are insulin sensitive? That is GLUT4. GLUT4. GLUT4 is insulin sensitive. If insulin is there, GLUT4 will be present in the membrane. Classically, GLUT4 are present in, yes, let me show you that GLUT4 is present in muscles and in the adipocytes. Suppose this is skeletal muscle, right? Here it is myocardial muscle, and here is a fat cell. Fat cell. Now, GLUT4 are not normally expressed on the cell membrane. GLUT4 are normally present inside the cell on special vesicles. They are inserted on special vesicle. This is GLUT4. When insulin signals come, GLUT4 moves to the membrane and insert into membrane, and in this way a part of the membrane expresses which channels? GLUT4. GLUT4. So GLUT4 are normally not present on the target tissue. But if insulin is present, right, only then from intracellular location GLUT4 will insert themselves into the cell membrane. So these GLUT4 are, or their presence in the cell membrane is insulin dependent, and these insulin dependent GLUT4 are also present in adipocytes. In adipocytes, what are these? GLUT4. So I wanted to compare and contrast that even though the major organ on which insulin work is liver, skeletal muscle, of course muscle mean skeletal muscle and cardiac muscle, and adipose tissue. Is that right? The major organ on which insulin has major effect is your liver, muscles, I will say here is your liver, It has action on liver, insulin. Insulin has action on muscles and has action on adipocyte. These are the main target tissue. But I want to tell you that liver has what kind of GLUT? It has GLUT2. GLUT2 are not insulin dependent. So insulin on hepatocyte alters many enzymatic pathways, right? Insulin alters many enzyme functions inside the hepatocyte but does not change the permeability of the membrane to glucose. Hepatocyte permeability of the membrane to glucose remains all the time. It is insulin independent. Insulin also work on muscles and adipocyte. Here also it lead to changes in metabolism, but also here it changes the cell membrane, and cell membrane under the influence of insulin will express GLUT4, because these GLUT4 only move to the membrane when insulin is present, and if insulin is catabolized, these GLUT4 will move in again, right? Membrane will not be able to take up glucose. So we can say GLUT4 in the muscles and in the adipocyte are insulin dependent, but GLUT2 wherever they are present, they are not insulin dependent, right? They are insulin independent. So uptake of glucose by the liver is insulin independent, but insulin has other function on the liver. But on the muscle and the adipocyte, insulin has metabolic function as well as function of increasing the uptake of glucose or making the membranes permeable to glucose. Is that clear? Now we come back. So what will happen, that here is your beautiful glucose molecule, right? When it is passing by, as from gastrointestinal system, very high concentration of glucose is coming, and GLUT2 are two ways. They are two ways, from high concentration to low. In the fed state, when you have eaten a lot of food, lot of glucose is coming in portal circulation. So glucose will enter into hepatocytes, right, through which GLUT2, right? Once the glucose enter, now listen carefully, now we have to see what happens to this glucose. Glucose has entered into cell. If glucose stay here, very rapidly an equilibrium will be achieved and more glucose cannot be taken. But liver cell want to utilize this glucose and store this glucose or convert glucose into such molecule which can be sent to other tissues where they can be stored. So what will happen, that within liver a special type of enzyme is here which is called glucokinase. What is this enzyme? Glucokinase. Now actually glucokinase will be activated, which will act on the glucose and convert the glucose into glucose 6-phosphate. It will convert the glucose into glucose 6-phosphate. It means that this enzyme has led to the phosphorylation of glucose at the carbon number 6. Now where was this enzyme before the glucose come? It's very interesting. Previously this enzyme was present in the nucleus of the hepatocyte. This enzyme was present in the nucleus and it was kept there by a special regulatory protein, right? This regulatory protein catches the glucokinase. So this protein is called glucokinase regulatory protein. Now in the hepatocyte, there are glucokinase regulatory proteins. If glucose is not coming, this regulatory protein drag the glucose kinase and pull it into nucleus and terminate the function of glucokinase. But as soon as glucose start entering, you know what glucose does? First glucose enter into cell, pass through the cytosol. moves through the cytoplasm and pass through the nuclear membrane and modify the glucose kinase regulatory protein in such a way that glucokinase is set free and glucokinase come out. What did I say? That once glucose start entering, it will not only enter into cytosol, but it will also reach to the nucleus, and from there it will help the glucokinase to get rid of glucokinase regulatory protein, and this will come over here, glucokinase, and it will convert glucose into glucose 6-phosphate. Secondly, very important, insulin RAS-MAP kinase pathway also, this pathway also activates the gene which lead to synthesis of this enzyme. You are understanding? So this enzyme activity, what is this enzyme activity? Glucokinase activity. This is increased after taking food, because today's lecture is not only about insulin, not only defenses against hyperglycemia. I'm also teaching within that and integrating all the biochemical pathways related with the fed state. And later on we'll talk about fasting state also and preventing hypoglycemia. So that once we start diabetes mellitus, our foundations are perfect. Come back. So what I'm saying, that glucokinase activity is increased by two mechanisms. Number one, glucose goes into nucleus and alter glucokinase regulatory protein in such a way that glucokinase which was preformed, that is released from there and appears here. Number two, insulin gives signal to the nucleus and gives order to synthesize more glucokinase, right? Now, what glucokinase will do? Glucokinase will convert glucose into glucose 6-phosphate. Now, glucose 6-phosphate is a product in metabolic map. Glucose 6-phosphate is at a very special situation, that it can move in many directions. I will explain it. It can move upward and can be converted into glycogen. It can move forward further and enter into, okay, this is glycogen pathway, glycogenesis. Glucose 6-phosphate can be mobilized to convert into, what is this? Glycogen. Or glucose 6-phosphate can be pentose phosphate pathway, right? It can go to that pentose phosphate shunt, which was previously called hexose monophosphate shunt. Or glucose 6-phosphate can go down and go under the pathway of glycolysis. So we can say, or in the liver, yes, in the liver, in fasting state, when blood glucose level is going down, an enzyme is activated which will convert glucose 6-phosphate back into glucose, and glucose will go out. So it means that glucose 6-phosphate is a metabolite which is present in the metabolic map at a very special position. If I am glucose 6-phosphate, I can move upward, and many glucose 6-phosphate can be utilized to make glycogen. I can move forward, and what I can do? I can go hexose monophosphate shunt pathway. I can move backward and go out of the cell by dephosphorylation pathway, or I can go downward and I can enter into glycolysis pathway. So this is a special type of molecule which is, you can say, a very, very important, and what will determine in which direction it will move, it depends on enzymatic activity and availability of glucose 6-phosphate. In fed state, when blood glucose is high, glucose is coming in and glucose is acted upon by glucokinase, and it is glucose is converted and trapped in the form of glucose 6-phosphate. Now glucose 6-phosphate cannot go out, because due to attachment of phosphate it becomes very heavy and heavy polar, and it cannot pass through glucose transporters, right? But if fasting state occur, listen now carefully, in fasting state, when you are very hungry or might be angry too, you have not eaten for many hours, you know what happens? This other enzyme is activated and glucose 6-phosphate will convert into glucose. It means phosphate will be removed. So this enzyme is called glucose 6-phosphatase. What is it called? Glucose 6-phosphatase. Now we have to remember a very important point, that liver, hepatocyte have both kind of enzyme. It has glucokinase, also it has glucose 6-phosphatase. In fed state, this is active. In fasting state, this is active. So in fed state, liver is taking up the glucose and storing it and utilizing it. In fasting state, stored glucose is broken down, and because liver cells have glucose 6, what is this? Phosphate, it can set the glucose free and provide the glucose to the blood. It means, again, it is like a mother. When extra glucose is coming, it is storing. And when glucose in the blood level is going down, the liver converts into glucose producing organ. And one of the important reasons why liver is able to release glucose, because liver has glucose 6-phosphatase and it can convert glucose 6-phosphate into simple glucose and let it out. Am I clear? Now, of course, under present circumstances, which we are talking about, that there is fed state, in this fed state, what I am saying, that this enzyme is active, this enzyme is in active state, glucokinase, and this is in inactive state, right? Why this is active? Because glucose itself brought it from the nucleus, dragged it out from the claws of glucose kinase regulatory protein. Secondly, insulin was able to signal the nucleus to synthesize more glucokinase. But at the same time, insulin gives signal, yes, insulin gives signal to the nucleus that the gene which produces this phosphatases, that gene should be inhibited. It means insulin can lead to induction or stimulation of the genes which produce glucokinase, and insulin lead to inhibition of the genes which produce glucose 6-phosphatase. In this way, insulin favors that more and more glucose will enter. Why? Because more and more glucose will convert into glucose 6-phosphate and free glucose in the cell will be less. And when free glucose in the cell will be less, then from more glucose which is outside, concentration gradient will move from out to in. In this way, hepatocytes will take up the glucose through GLUT2. Now, once the glucose under the influence of glucokinase has been converted into glucose 6-phosphate, what will happen to glucose 6-phosphate? One thing is, it simply should go to glycolysis pathway, Krebs cycle, and then it goes to the electron transport chain and produce ATP and energy for the cell, right? So very initial glucose which come, that can be used to produce ATP. For that purpose, it should go through the glycolysis pathway and then Krebs cycle and then into electron transport chain and convert ADP into ATP, and energy level of the hepatocyte are met. But glucose is too much coming. Now what to do with the extra glucose? What to do with the extra glucose? Then it will use another pathway which is upgoing pathway. Here is glycogen house. What is this? This is glycogen storage house, glycogen store. Now what happens? Actually, through multiple steps, glucose 6-phosphate, right, then it will convert into glucose 1-phosphate and then UDP and many pathways. Eventually, these glucose molecule will polymerize. What will happen? These molecules will polymerize. Polymerize means that if there was, for example, this is 3 glucose molecule, if they become sticking like this, we say there's formation of polymer. Is that right? Now, extra glucose, which is more than the need of the hepatocyte energy requirement, extra glucose will be acted upon by special enzymes. The enzyme which is present over here, that is, yes, what is the name of this enzyme? Glycogen synthase. What is the name of this enzyme? Glycogen synthase. Glycogen synthase. Now, glycogen synthase, and there are many other enzymes on the way, but it is the best regulated enzyme in this pathway. Glycogen synthase is able to convert glucose 6-phosphate into glycogen and store the glucose in the form of polymers of glycogen. Now, it means insulin should stimulate the glycogen synthase. When glycogen synthase is phosphorylated, it is inactive. It is inactive. So, if insulin wants that more and more glucose 6-phosphate should convert into glycogen, then insulin should give signals in such a way that glycogen synthase should be activated. But when glycogen synthase is phosphorylated, it is inactive. To activate this, some insulin should activate some phosphatase which should remove this phosphate. And when this phosphate is removed, we say this enzyme is dephosphorylated. And as soon as it is dephosphorylated, it becomes happy. It becomes happy and start the full action and rapidly start converting what? Glucose into glycogen. Is that right? Question is that why glycogen is a very good form of glucose storage, why glucose cannot be stored as such? Remember, if, for example, there are 5 molecules of glucose here and there are 5 molecules of glucose which are united together into a chain. Now, this is polymer of 5 glucose molecules. This is one polymer. Its total osmotic pressure will be less. But if these remain independent, separate, if all the glucose which is coming and it remain as individual molecules, they will produce so much osmotic drag that hepatocyte will pull a lot of water and burst. Do you think it should happen? So, when a lot of glucose is coming, hepatocyte has to find a way to store this glucose. But do not keep the glucose as individual molecules, because osmotic power does not depend on what is the size of the molecule, it mainly depends on the number of the molecules. So what happens that nature has devised a way that glucose 6-phosphates are under the directions of dephosphorylated glycogen synthase convert into glycogen polymers and store into glycogen house. Is that right? Now, here I want to mention one more thing. Here, dephosphorylation occurs and glucose is polymerized into glycogen polymers. Is that right? Dephosphorylation of glycogen synthase occurs. But at the same time, there is another pathway in which glycogen can be broken down and brought back to glucose 6-phosphate. For example, in fed state, when you have eaten, in absorptive state, when a lot of glucose is coming to the liver should use it pathway upward and store the extra glucose as glycogen. But in fasting state, when there is a risk of hypoglycemia, this glycogen should be broken and brought to glucose 6-phosphate. This should be inhibited and phosphatase should be activated and let the glucose out. But you understand. So there is a pathway going upward, that is glycogenesis. And there is a pathway from here coming downward, that is glycogenolysis. So what really happens? That insulin in the hepatocytes and also in the muscle, it increases, stimulates the glycogenesis and inhibits the glycogenolysis. Again, what happens? That in fed state, number one, insulin will stimulate the glycolysis for energy pathway and some other purposes which we will discuss later. But insulin also stimulates the glycogenesis, glycogen formation, and it should, in fed state, when there is abundance of glucose, right, insulin should do something to the enzymes, that those enzymes which are involved in breaking down of the glycogen, they should be inhibited. Is that right? If insulin has to work intelligently, then whenever more glucose is coming, it should be able to stimulate the pathway for glycogen formation and inhibit the pathway for glycogen breakdown. Now, the two key molecules which are involved in glycogen breakdown into glucose 6-phosphate, those two key enzymes are, yes, glucose, one is glycogen phosphorylase kinase and other is glycogen phosphorylase. Now listen, these two enzymes, when they are activated, glycogen phosphorylase kinase and this is glycogen phosphorylase. Glycogen phosphorylase actually removes the glucose molecule from the glycogen polymers, right? And eventually cell has a lot of glucose 6-phosphate and then glucose will go out. Now these two are very funny. You know what is the funny thing about them? That when they are dephosphorylated, they become inactive. It means what insulin has to do? Remove the phosphate of all three of them, dephosphorylate the glycogen synthase and it becomes active, and dephosphorylate the enzymes which were related with glycogenolysis, and dephosphorylation actually inhibits these enzymes. Am I clear, or you look confused? Are you? Okay, let me explain it. First of all, you have to understand that if let's suppose I'm standing here, right? I'm glucose 6-phosphate. Is it right? If I move upward, I convert into glycogen. And the process is called glycogenesis. Is it right? And if glycogen is broken down and eventually glucose 1-phosphate and 6-phosphate is released, and eventually then glucose is released, now breakdown of glycogen is called glycogenolysis. In fed state, when you have eaten a lot, you want to form the glycogen or you want to break down the glycogen? You want to form the glycogen. So if you want to form the glycogen, then glycogen synthase should be stimulated and glycogen breaking enzyme should be inhibited. But the beauty is this, that to stimulate this, it should be dephosphorylated. And to inhibit these should also be dephosphorylated. I told you, some enzymes, when they are dephosphorylated, they become active. And some enzymes, phosphate is at the top, right? In dephosphorylated state, they become inactive. So what I'm trying to say, that basically insulin activates many pathways, including pathways of phosphatases, which remove phosphate from many enzymes. And insulin gives signals for many enzymes to be dephosphorylated. And many enzymes, when they get dephosphorylated, they convert into active state. But there are exceptions, that there are some enzymes when they are phosphorylated, they are activated. Here what I'm saying, that glycogen forming enzymes, when they are dephosphorylated, they become active. glycogen breaking enzymes, when they are dephosphorylated, they become inactive. Now I said glycogen forming enzyme is glycogen synthase. Mainly, there are others also, but regulated step is glycogen synthase. And glycogen breaking enzymes are mainly glycogen phosphorylase, enzyme glycogen phosphorylase, and enzyme which add a phosphate to glycogen phosphorylase, that is glycogen phosphorylase kinase. You understand it? So what really happens, that when we want to break down the glycogen phosphorylase kinase become phosphorylated, it become active, it add a phosphate here and it become active, and both of them help in breakdown of glycogen. But in fed state, when glucose level is high, a lot of glucose is being absorbed from the GIT, and this is threatening to produce hyperglycemia. This is the time to store the to make the glycogen, not to break the glycogen. So I will sum up this. Glucose enter through GLUT2. It is acted upon by glucokinase, right? Activity which is increased, right? And glucose keep on converting in glucose 6-phosphate, right? Some of this glucose is used in glycolytic pathway and Krebs cycle and electron transport chain to produce energy. So cell will, first initial molecule will be used to fill up the energy store of the cell. If extra, what comes? If extra glucose come, then that glucose 6-phosphate will be polymerized. Glucose molecules will be actually polymerized into glycogen, and glycogen formation pathway or glycogenic pathway will be stimulated. But opposite pathway, which is glycogenolytic pathway, that will be inhibited. Glycogenolytic pathway will be inhibited. To stimulate this and inhibit this, actually mechanism is same, dephosphorylation of the enzymes. You dephosphorylate the glycogen synthesis, they become active. You dephosphorylase enzymes, glycogen phosphorylase kinase and glycogen phosphorylase, these are glycogenolytic enzymes. When they are dephosphorylated, they are inhibited. I hope now you are clear, otherwise I will commit suicide. Sure, I don't need to commit suicide. Okay, thank you. So now we come, let's suppose more glucose is coming, right? What to do with it? More glucose will go to another pathway, and this pathway in the end will end up into ribulose. We call it hexose monophosphate shunt, or this is called pentose phosphate pathway. I will not go into detail of this, but I am saying that some of the glucose 6-phosphate will be influenced by another group of enzyme, and that enzyme name is glucose 6-phosphate dehydrogenase. Glucose 6-phosphate dehydrogenase, right? What is the name of this enzyme? Glucose 6-phosphate dehydrogenase. And this enzyme will activate a pathway which is basically called hexose monophosphate shunt pathway in my times. In your modern times, we call it pentose phosphate pathway. What is the advantage of this pathway? This pathway basically convert NADP into NADPH, right? Now this is low energy molecule, this is high energy molecule, right? So hexose monophosphate shunt or pentose phosphate pathway, right, once it is activated, this will convert NADP plus into NADPH. So it will be high energy molecule. And later on in the lecture, we will see this NADPH will be utilized for the synthesis of fatty acids, right? This NADPH will be later on used for the synthesis of fatty acid. Now, glucose 6-phosphate dehydrogenase, this is allosterically regulated by NADP. The more NADP, what is this? More NADP bind with this enzyme become active and convert the NADP into NADPH. Secondly, glucose 6-phosphate dehydrogenase is also allosterically stimulated by the substrate. What is the substrate? Glucose 6-phosphate. So we can say this enzyme is regulated by allosteric mechanisms, not covalent mechanism, not by phosphorylation, dephosphorylation. Is that right? Am I clear? Yes. So what we have seen up to now, glucose enters into cell, glucokinase converted into glucose 6-phosphate. The choices are, number one, glucose 6 pathway go to glucose 6, enter into pathway to produce energy, or then extra will go to make the glycogen formation, more extra will go to make enter into pentose phosphate pathway and convert NADP into NADPH. Is that clear? Any question up to this? There's no. Now if still more glucose is coming, what to do now? If glycogen store become full, glycogen stores are full, you cannot store and you cannot convert more glucose 6-phosphate into this, and NADPH is also available in good concentration. Now what to do with the extra glucose? Now again it will drag that glucose along the glycolysis pathway, glycolytic pathway, which in the end produces pyruvate. There are many steps, right? And eventually this produces glucose 6-phosphate, right, which is 6 carbon unit. It will be broken down by the glycolytic pathway into 2 molecules of pyruvate, and each molecule of pyruvate is 3 carbon. This was 6 carbon unit, it will be broken down into 2 molecules of 3 carbon, 2 molecules of pyruvate. Now, here I want to show one thing, that some of these molecules which are going down, on the way they will convert into 3 carbon unit, and this carbon unit is called glycerol. Later on I will tell you that this glycerol will be also used for fatty acid synthesis. Actually, you know, when you take too much sugar, liver converts that sugar into fat. First, little bit glucose utilized for energy for the cell. Then some of that stored in, what is this? Glycogen formation. Then some of them are utilized to get ready to synthesize fatty acid and triglyceride, that is storage form. For fatty acid synthesis, we need NADPH as an energy. So this is made. For fatty acid synthesis, what is fatty acid? Fatty acid synthesis and triglyceride synthesis, we need one glycerol. This is one glycerol, right, with 3 fatty acids. This is triglyceride. What is triglyceride? Glycerol backbone with 3 fatty acids. So actually, when too much glucose is coming, glycerol is also being formed. So energy for fatty acid synthesis is also formed. And what is this? Glycerol, which is the backbone of triacylglycerol, that is also formed. And now we have to make fatty acid. How to make fatty acid? Actually, this glycolytic pathway should go. Now glycolytic pathway has 3 enzymes which are regulated, well regulated enzyme. One enzyme was glucokinase, right, taking the glucose and converting into glucose 6-phosphate. Then another enzyme on the way is, another regulated enzyme on the way is phosphofructokinase 1, right? This is also regulated enzyme, and this phosphofructose 1, this is positively regulated by insulin, right? And it is positively regulated by deficiency of energy. If cell has high levels of AMP, it means ATP is converted into ADP and ADP into AMP. So AMP can stimulate it. In the same way, insulin can also increase the synthesis of this enzyme. Plus, it is allosterically regulated by fructose 2,6 bisphosphatase. Forget about that. You just trust me that when a lot of glucose is coming, right, and glycogen stores are full, glycolytic pathway become very active. Glycolytic pathway become very active. Because we will see, if glucose moves on the glycolytic pathway, will pass the glucose through certain pathways and end up with a fatty acid. And you become fat. Right? So, what we have done, one regulated enzyme was glucokinase for glycolysis, other regulated was phosphofructokinase. Synthesis of this is also increased by insulin, synthesis of this is also increased by insulin, and it is also stimulated allosterically by increased AMP, which signals the deficiency of energy, and it is also activated by fructose 2,6 bisphosphatase phosphate, but I will not discuss in detail that thing. Now, after that, another enzyme which is well regulated is pyruvate kinase, right, which convert the phosphoenol pyruvate into pyruvate. This enzyme is also affected by insulin. Insulin increases the synthesis of this enzyme, right, and this enzyme is also activated by dephosphorylation. Dephosphorylation, its phosphate is killed. This was also activated by dephosphorylation, but these were inhibited by dephosphorylation. So dephosphorylation was increasing. What was happening? Up to now, insulin has done a lot of dephosphorylation. It dephosphorylated the enzyme in glycogen formation and activated them. In dephosphorylated the enzyme which was what? Glycogenolytic enzyme, and they were inhibited. It dephosphorylated the, what is this? Pyruvate kinase and stimulated it. Right? So it is also regulated. So how many enzymes? 3 enzymes I am showing which are regulated: glucokinase, phosphofructokinase and pyruvate kinase. All of them are positively affected in the presence of insulin and presence of lot of glucose. Now this pyruvate will enter into, yes, what is it? Mitochondria. What is it? Mitochondria. This pyruvate will enter into mitochondria, and here is enzyme in the mitochondria, inside the mitochondria, but I will draw it outside. Enzyme, that enzyme is called pyruvate dehydrogenase. That will convert pyruvate into, yes, that will convert pyruvate into acetyl-CoA. Right. So we ended up with acetyl-CoA. Again, listen. Pyruvate, and there are 2 enzymes. One is pyruvate kinase, other is pyruvate dehydrogenase. Pyruvate kinase is in cytosol and it helps in formation of pyruvate. And pyruvate dehydrogenase is inside the mitochondria and it helps in converting pyruvate into acetyl-CoA. Right? Now, pathway from glucose up to pyruvate was glycolysis. What was that? Glycolysis. Then pyruvate enters inside, and once pyruvate enter inside what? Mitochondria. It is attacked by pyruvate dehydrogenase and it converts into acetyl-CoA. Now, pyruvate is 3 carbon unit, acetyl-CoA is 2 carbon unit. It has lost 1 carbon. Now, acetyl-CoA, you must be knowing that if you talk about the Krebs cycle, acetyl-CoA fuses with oxaloacetate. Oxaloacetate and acetyl-CoA, they fuse with each other. Now, oxaloacetate has how many? Oxaloacetate, okay, I will draw it here. Oxaloacetate, right, how many carbon? 1, 2, 3, 4. Acetyl-CoA, it has how many carbon? 2. It has 2 with 1 CoA. Now what happens? Oxaloacetate and acetyl-CoA become together and CoA is removed. When they become together, they unite with each other, they end up with how many carbons? 6. And this 6 carbon unit is called citrate. What is it? Citrate. It is not glucose because here carbons are arranged in a different way. So we call it citrate. Am I clear? Now we have taken the glucose from here. Look at the naughty hepatocyte, what it is doing with this glucose. It is using this glucose to fill up the stores of glycogen, using this glucose to move to pentose phosphate pathway to make NADPH, so that in future it can use it for energy source as for fatty acid synthesis. On the way, when it makes the glycolysis too much, with some glycolysis and Krebs cycle, it makes energy and uses for itself. Then more glucose come, it still super activate the, what is this pathway? Glycolytic pathway. On the way, some of the metabolite of glycolytic pathway in mid pathway will convert into glycerol. That glycerol will in future help in formation of triglycerides. Then eventually glucose 6-phosphate will break down into 2-pyruvate. Now this pyruvate will enter into cell and convert into acetyl-CoA. Acetyl-CoA has 2 carbon and oxaloacetate has how? 4 carbon. These 4 carbon plus 2 carbon, they become united and CoA is lost. But the new molecule which is made is called citrate. It is called citrate. Okay, let me make mitochondria a little more longer. Hepatocyte, and here is mitochondria. Now, so acetyl-CoA, so what is happening? Entering glucose here is leading to lot of formation of acetyl-CoA, right? Now acetyl-CoA is converted into citrate. Citrate will convert into isocitrate, right? And isocitrate convert into alpha ketoglutarate, right? It should be still inside the mitochondria. Alpha ketoglutarate. And then Krebs cycle continues. But I want to tell you something which is very important. If it completes the Krebs cycle, then it will help in formation of ATP. We don't want any more ATP, or do you want? No. So Krebs cycle should be stopped somewhere. Now the problem is glucose is coming, it means acetyl-CoA is coming, right? And citrate is coming, isocitrate is coming. But in the cell, level of ATP go up, AMP level go down. It means molecular signals are that cell is very rich in energy. Hepatocyte is very rich in energy. Why? Because ATP already made a lot and most of the AMP convert into ADP and then into ATP. So AMP level low. Now low level of, here it is citrate, then isocitrate. Should I make it here? Okay, let me make the cycle here so that you really see that what's going on. I will rapidly draw what we have learned there, right? Now you will tell me, this is a glucose molecule. It entered through which GLUT? GLUT2, very good. Through the GLUT2 it entered, and this glucose molecule become glucose 6-phosphate by the action of? Yes, by the action of glucokinase, okay? And the enzyme below which could reverse is inhibited. then this glucose 6-phosphate will go to energy pathway. Also, it will lead to glycogen formation, right? In this case, there was dephosphorylation of synthases, but that enzyme was activated. And there was also this pathway enzyme, glycogenolytic, they were also dephosphorylated, but dephosphorylated has opposite effect here and lead to inhibition of this enzyme, right? After that, we discussed glucose 6-phosphate also can be converted into, it can go to pentose phosphate pathway, and during this it produces NADPH, and this is a source of energy which can be later utilized into fatty acid synthesis. Then glucose 6 pathway can come down. Up to now, insulin has increased glucose uptake, but not by inserting the GLUT2, by converting the glucose into glucose 6-phosphate and keeping the intracellular glucose concentration low so that external glucose can come in. Insulin has increased the glycogenesis. Insulin has decreased the glycogenolysis. Insulin has activated the pathway, pentose phosphate pathway, in such a way that there is more formation of NADPH, which will be later on used to synthesize fatty acid as an energy source. Then insulin, of course, stimulated the pathway which was glycolytic pathway, and pyruvate was made. Pyruvate, in somewhere in the middle, what was there? What was this made? Glycerol was also made. Is that right? Now, this is the energy for the fat synthesis. I should highlight. And this is the backbone of triglyceride. But we still have not made fatty acids, right? Now, we can, pyruvate will enter into what? Mitochondria. I will make a mitochondria here. Pyruvate will enter into mitochondria. There, pyruvate will convert into acetyl-CoA, and acetyl-CoA will fuse with oxaloacetate, and oxaloacetate will convert into 6-carbon citrate, and then citrate will convert into isocitrate, and that convert into alpha ketoglutarate, and so forth, and cycle is completed. But let's take a break for 10 minutes, and then I will tell you that from this citrate, yes, we have to stop this cycle and use this citrate to make the fatty acids. How we do that? After the break. Hello everyone. So we continue our lecture, that now we were talking about that hepatocyte has already utilized some of the glucose for energy, its own energy purpose. Some of the hepatocyte, some of the glucose has been used to store as glycogen, rather a lot of glucose. Then some of it has been used by monophosphate shunt to produce NADPH. From the glycolytic pathway, we have also made the glycerol backbone. Now, too much glucose which was coming, we are taking it through the glycolytic pathway and then into Krebs cycle. And let's play something and convert that into fatty acid. How we can do that? We come over here. That this was your acetyl-CoA, right? When you have eaten lot of carbohydrates, right? We say when you take lot of Coca-Cola full of sugar, or you take lot of candies, they will be added here. How come? Glucose molecule is converted into fat. That is what now I am telling you, that is one of the master function of the liver, right? That up to now you understand, acetyl-CoA is made, acetyl-CoA with oxaloacetate, right? Both of them fuse together and make what? Citrate. And citrate is 6-carbon unit, right? Citrate can be converted into isocitrate and isocitrate into alpha ketoglutarate. Now, if the cycle keep on moving, too much energy will be produced which is not required. So what to do with this extra citrate? We have to stop the Krebs cycle somewhere and force the intermediate of it to convert into fatty acid. How nature does it? Nature does it by putting a block here. Putting a roadblock here does not allow isocitrate to convert into alpha ketoglutarate. Now the enzyme which is present over here, that is what? Isocitrate dehydrogenase. There is an enzyme here present, isocitrate dehydrogenase, and this enzyme should be inhibited so that isocitrate cannot convert into alpha ketoglutarate, and we put a block for energy production. We put a block for energy production, and then we will convert this citrate, because once we put a block here, isocitrate and citrate are interchangeable, and we will convert this citrate into eventually fatty acids. But how? Before that, I would love to explain that how isocitrate dehydrogenase is inhibited. Very simple, very simple. It should be inhibited when there are cues for high energy level in the cell. For example, what should inhibit it? It should be inhibited by high ATP. It should be inhibited by, yes, it should be inhibited by high level of NADPH, right? It should be inhibited by its product accumulation of alpha ketoglutaric acid, this one, right? Now, all of them are allosterically regulating and inhibiting. They are, we can say, all of them are high level of ATP in the cell, high level of NADPH in the cell, high level of alpha ketoglutaric acid here. All of them negatively regulate allosterically isocitrate dehydrogenase and inhibit it. But there is one more thing. Once isocitrate dehydrogenase is inhibited, now mitochondria start accumulating what? Citrate. Start accumulating citrate. Now what to do with this citrate? Citrate should get out of the mitochondria. Citrate should get out of the mitochondria. So there is a special transporter here. There is a special transporter here, and we call it citrate transporter. Citrate transporter or citrate shuttle, which will get the citrate outside the mitochondria, because fatty acid is synthesized in cytosol. Fatty acid is synthesized in cytosol. So what happens, that citrate shuttle or citrate transport system, once isocitrate dehydrogenase is inhibited, citrate accumulate into mitochondria, start going out. Once the citrate goes out, then what happens? Okay, now citrate is out. Citrate will break down into 2 carbon unit and 4 carbon units. It was 6 carbon. With this 2 carbon unit, it will have CoA. What is it? CoA. So what is it? Acetyl CoA. Now this acetyl CoA is cytosolic acetyl CoA. Previously we had acetyl CoA, which was intramitochondrial acetyl CoA. Intramitochondrial acetyl CoA runs the Krebs cycle and produces energy. But when you find CoA outside the mitochondria into cytosol, it is going to help in formation of fatty acids. And citrate break down into acetyl CoA, and what is this? This is, yes, this is oxaloacetate. What is this? Oxaloacetate. And this oxaloacetate will convert into malate. I will not go into detail of this, and malate shuttle will take it back. Malate shuttle will take the malate into where? Mitochondria, and that will be used somewhere else. We'll talk later. Now, the thing is that the master thing which we did, that we got the citrate out through the shuttle and we provided the cytosol with the acetyl-CoA. Right? Now, in whole this thing, insulin has played a lot of role. As I told you, insulin increases this cell glucokinase activity. Insulin also alters the activity of different pyruvate kinases and dehydrogenases, and because a lot of energy was produced, isocitrate dehydrogenase was inhibited, and when this Krebs cycle stopped, citrate escaped out through citrate transporter. This citrate was broken down by a special enzyme, but this is not important enzyme, that is called citrate lyase. That enzyme breaks down the citrate into acetyl-CoA and oxaloacetate. Oxaloacetate goes back by taking the cover of malate and re-enters the mitochondria. In this way, more and more glucose is coming and more acetyl-CoA is going to cytosol. What is happening now? This is the beginning of fatty acid formation. Actually, this acetyl-CoA, what is this? Acetyl-CoA will be acted upon by another enzyme, and this enzyme is very important. This is ACC, acetyl-CoA carboxylase. What is it? Acetyl-CoA carboxylase. Now this acetyl-CoA carboxylase, right, this is very important enzyme. There are only two important enzymes in formation of fatty acid, and this is more important out of those two. Now this acetyl-CoA carboxylase, this basically add a carbon unit to this and convert into 3 carbon unit, and that with what is this? CoA. So we call it, this was acetyl-CoA, and this is malonyl CoA. So in this way, malonyl CoA is formed, right? And this was done by which enzyme? Acetyl-CoA carboxylase. Now, what are the regulators of this? Number one, citrate itself. Citrate, more and more citrate appear in cytosol, more and more this enzyme is activated. This is an example of what kind of stimulation? Allosteric regulation, allosteric positive regulation. So more citrate come here, more acetyl-CoA carboxylase is activated and converting the acetyl-CoA into malonyl CoA, number one. Number two, insulin lead to dephosphorylation. This phosphorylation is broken. Insulin lead to dephosphorylation of this enzyme, and when dephosphorylation occur, that will also leads to activation. Now we have learned that many enzymes by dephosphorylation can be activated, but some enzymes can be inhibited by dephosphorylation. You remember, glycogen synthase was activated by dephosphorylation, but glycogen breaking enzyme were inhibited by dephosphorylation. Then many enzymes on this pathway by dephosphorylation were stimulated, and here now we have reached outside that ACC, that is acetyl-CoA carboxylase, which convert acetyl-CoA into malonyl CoA, that is also stimulated by dephosphorylation. Then another thing, insulin also goes to the genes of the cell and stimulate the genes and increase the synthesis of acetyl-CoA carboxylase, right? So it means that this is very well regulated enzyme, that more citrate, more action of ACC, right? More dephosphorylations, more action of ACC, right? Insulin act on RAS-MAP kinase to the nucleus, more production and action of ACC. So in these cells, when you take too much sugars or you are providing too much glucose, then actually glucose derivatives will take very unusual pathway. That initially this glucose derivatives to make the fatty acid, they have gone to, what was this, pentose phosphate pathway, made NADPH, which was energy required for what? Fatty acid synthesis. Then glycolysis was accelerated, and on the way it made backbone of glycerol. Then pyruvate went in, acetyl-CoA in the cytosol, citrate, isocitrate, but isocitrate dehydrogenase inhibited by high energy, and then citrate had to escape out through the transporters. When it runs out into cytosol, their citrate is taken up by the lyase and broken down into acetyl-CoA and oxaloacetate. This acetyl-CoA is acted upon by ACC, acetyl-CoA carboxylase, and can make malonyl-CoA, right? Now after that, the next enzyme is fatty acid synthase. What is this enzyme? Fatty acid synthase. Now this enzyme is fatty acid synthase, right? Now this enzyme will work on malonyl-CoA and work repeatedly and keep on adding the carbon units on that and elongating this chain. So it was 3 carbon, then 2 more carbon added. How much it become? 5, then more 7, then 2, then more carbon. We call it chain elongation process. So what happens once malonyl-CoA is formed and this 3 carbon unit is formed, then repeatedly fatty acid synthase complex keep on adding more and more carbon. From where these carbons are coming? These carbons are coming from acetyl-CoA. These carbons are coming from acetyl-CoA. These were the original carbons which were coming from glucose. And now these carbons are incorporated into fatty acid chain formation. Now these carbons are incorporated in formation of fatty acids. So when this chain will become quite long with multiple, eventually a fatty acid will be, fatty acyl-CoA will be formed. We call it fatty acyl-CoA, and eventually fatty acid is formed, right? So we come over this place and just repeat it, that we were saying there was citrate, there was isocitrate, there was alpha ketoglutarate. Enzyme which was putting a block here was citrate, yes, dehydrogenase, right? That was able to sense the high level of energy and got inhibited, and high level of alpha ketoglutarate. Then citrate went out. You remember that? Citrate went out and citrate become, okay, citrate is here out. I'm repeating it just very rapidly. Oxaloacetate, then into malate, and then back through malate shuttle. And here is which shuttle working? Citrate shuttle. Citrate break down into oxaloacetate and acetyl-CoA. Then what was the enzyme here? ACC. ACC was activated by citrate. It was activated by dephosphorylation, and its synthesis was also increased by the insulin, right? And then ACC, acetyl-CoA carboxylase, act on the acetyl-CoA and adds 1 carbon more, and it becomes 3 carbon unit, and this 3 carbon unit with CoA become malonyl-CoA. Here it was acetyl-CoA. Malonyl-CoA acted upon what? Repeated steps of, what was this? Fatty acid synthesis and fatty acyl-CoA, and then CoA is removed and this is fatty acid. In this way, the original glucose which was entering here ended up into fatty acid, right? Now it is so far, this fatty acid, and now during this lot of energy was required. That energy was provided by NADPH, which was coming from which pathway? Hexose monophosphate shunt. And now you use the glycerol backbone. This was your glycerol backbone, glycerol 6-phosphate, and you attach it glycerol, what is this, monoglyceride, now it is glycerol diglyceride, now it is glycerol triglyceride. In this way, eventually the carbons which were present in the glucose, they are used to make the glycerol backbone of the triglyceride, and also those carbons eventually were used to make the, what is this, triacylglycerol. And all this was orchestrated by mainly insulin. So what did insulin did in hepatocyte? Number one, it used the increase the uptake of glucose by converting rapidly glucose into glucose 6-phosphate. Number two, it increased the formation of glycogen, glycogenesis. Number three, it reduced the glycogenolysis. Number four, increased glycolysis. Number five, it made the Krebs cycle fast and produced a lot of energy. After enough energy, what did it do? Meanwhile, pentose phosphate pathway produced NADPH. Glycolysis on the way produced glycerol backbone, and now what was Krebs cycle gets stop, right? Because of inhibition of isocitrate dehydrogenase. And when that is inhibited, citrate goes out through citrate transporter, and citrate break down into oxaloacetate and acetyl-CoA. Acetyl-CoA acted upon by ACC and converted into malonyl-CoA with repeated addition of 2 carbon units. Eventually fatty acyl-CoA and fatty acids are formed. Then fatty acids are attached with the glycerol backbone, and that is converted into what? Triglyceride. Now this triglyceride, wow, liver cannot store so many triglycerides. So what liver will do? Liver will pack them as a gift pack. You know, this was your sugar, and now it is going to your abdomen and becoming fat. How? Liver will take this triglyceride and pack them into a special type of gift pack. I will make that gift pack here, gift pack, and there will be lot of triglyceride inside. Okay, these are triglycerides, and there are some other molecules also. I will not go into detail, but there are apoproteins attached, special proteins attached on it. There is apo B100, apo C1, and apo E. Now what happens, that triglyceride which are formed, right, special type of, what is this, lipoprotein is made. What is this? Lipoprotein, because it has lipids. It has a lot of triglyceride, some cholesterol esters, some phospholipids. So lot of, what is this, lipids are there, and with that some proteins are there. These are apoproteins, apo B100, apo C1, apo E. Now these are all lipids. With these proteins, we call it lipoproteins. What we call it? Lipoproteins. And because it is very rich in triglyceride, so its density is very low. It is not tightly packed. It's very fluffy. It is very fluffy. So this is called very low density lipoproteins, VLDL. VLDL. So these will be secreted out of hepatocytes. These triglycerides, recently made triglycerides, are secreted, released into blood again. So hepatocytes were taking the glucose, using it for energy, making more glycogen, and then making lot of fatty acids and glycerols and NADPH, and then eventually making triglycerides and packing the triglyceride into lipoproteins. Which kind of lipoprotein? Very loose and fluffy lipoprotein. We call it very low density lipoproteins. And In this way, in the blood, what will appear? VLDL. What is this? Very low density lipoprotein. From where they are coming? They are coming from liver. So liver is an organ that when you eat a lot, right, it takes the glucose, not only stores the glucose in the form of glycogen and uses it for energy, but if you are really eating too much, then all extra carbons of the glucose will be converted into carbons of glycerol and fatty acids, and lot of triglyceride will be made in the liver, and liver cannot hold so much triglyceride. Liver cannot hold so much, hepatocyte cannot keep so much triglyceride. So it keep on throwing the triglyceride packed in the form of VLDL. And this VLDL, later on we will see, will go to the adipose tissue and there it will be stored, right? But on the way, VLDL will go under some changes. But here I will stop the lecture, right? What I am saying, that basically liver is releasing VLDL, and in the VLDL there are triglycerides, and these triglycerides are endogenously synthesized. They are de novo synthesized, right? But triglycerides are also present in our food, and those triglycerides which are present in our food from the gastrointestinal system, they are enterocytes convert them into chylomicrons. Now VLDL is different and chylomicrons are different. Chylomicrons are also what? Lipoproteins. But chylomicrons are synthesized inside the enterocytes. Chylomicrons are synthesized in the mucosal cells of GIT, right? There, the triglycerides, which are exogenous triglyceride, they are packed into what? No. Gastrointestinal system and small intestinal mucosal cells, they make the chylomicrons and pack the exogenous triglyceride. So triglyceride which you eat, they are present in chylomicrons, and chylomicrons are synthesized where? In gastrointestinal cells. Is that right? And from there they enter into lymphatics. Chylomicrons enter into lymphatic pathways, lacteals, and then they go to thoracic duct, and eventually thoracic duct drain into venous system, and chylomicrons come into venous system. Now what did we learn? We learned this thing that triglyceride rich chylomicron, and okay, triglyceride rich lipoproteins can be chylomicrons or can be VLDL. Is that right? Now chylomicrons are coming from where? From GIT. VLDL is coming from where? From liver, hepatocyte. So chylomicrons are coming from enterocytes and VLDL is coming from hepatocytes. The triglyceride present in chylomicrons are exogenous, and triglyceride present in VLDL are endogenous, right? And in next lecture we will learn that both of these chylomicrons and VLDL go to different tissues where triglyceride can be extracted, broken down, fatty acids can be taken away and stored or utilized for energy. That we will be discussing in next webinar. After the break, we will go for the question answer session. Now, we are still talking about fed state. What we learned today, we did learn about insulin, but we also learn about fed state and role of the liver, and we also learn about many metabolic pathways and how they are influenced by insulin or allosterically, or enzymes are activated by other. But I have completed the work on hepatocyte. In next webinar, I will be working on anabolic actions of insulin, where? On muscles and on adipose tissue and their enzymes, right? And on the heart, and what is the relationship of the brain which is independent of insulin, right? We will discuss in next webinar, right? Now let's have a short break. After that we come to the question answer session, right? And please go through this repeatedly, because this is not only foundations for diabetes mellitus. As you know, diabetes mellitus is a bag of metabolic diseases. This is foundations for all your biochemistry and metabolic pathways and intermediary pathways. How do they work? Once we have done this fed state, it will be very easy to learn the fasting state, because not all but most step will be reversed, right? Okay, let's have a break and then we'll continue with your questions answer session.